It's a PDF, so it's like it's not working as a presentation.
Yeah, we already download. What makes it?
Yeah, I downloaded it like 3 times on accident.
Sorry.
Okay. Just
No, it's okay. You got it. Okay, now we're ready. Yeah. So our project is titled Using an AdEx Neural Network to Understand Impaired Gamma Oscillations in Psychological Disorders. So the main thing we're talking about, especially when it comes to like psychological disorders, mainly schizophrenia and some dissociative disorders, is NMDA dysfunction. And so we're going to be looking at different oscillatory bands, mainly gamma. So that's up in the top right. But yeah, it's been proposed that symptoms of schizophrenia stem from a lack of communication between areas, and a big, frequency for interareal communication is known to be gamma, or thought to be. So people with schizophrenia and other dissociative disorders exhibit altered gamma oscillations. So when it's spontaneous, it's more frequent, higher power gamma. And then when it's in response to like a specific stimulatory drive, they sometimes see less gamma. So it's kind of confusing and weird. In healthy controls, people have used ketamine to get some of the same symptoms as schizophrenia. And so the actual mechanism of action of ketamine kind of points to inhibitory interneurons and NMDA hypofunction for people with these disorders, as well as some GWAS data that seems to particularly affect inhibitory interneurons when compared to other cells. So that's going to be like our main target that we're focusing on. And so we based a lot of this on a paper by Alan Defects. He used 5,000 neurons in point neurons, AdEx, with a 4:1 regular spiking to fast spiking ratio. Each of them had a 10% chance to connect to another one. And then there was an external drive of 5,000 Poisson spike trains at 3 hertz. And then that also had a 10% chance to connect to any of the cells. And so they reproduced some observations that showed that NMDA receptor antagonists increase gamma oscillation power and frequency. Then they also saw some weird stuff with like, or not weird, it actually made a bunch of sense, but that with less inhibition or excitatory receptor activation on these inhibitory cells, there was more reactivity to external stimuli. And that happened in both gamma and asynchronous states.
Sorry about my voice, very bad. So in our project, we first aimed to build a neural model in SML that has AMPA, GABA, and NMDA channels, and then use this model to create a two-layer network of connected populations of inhibitory and excitatory neurons that resembles a cortical circuitry that can generate gamma oscillations using NEST. And finally, we changed the NMDA channel conductance to better understand the alterations that in cortical gamma that happens in schizophrenia and other disorders. So our first idea was to replicate the model from the paper on SML, but we did some alterations besides that. So it's an AdEx model of point neurons. Those are the main equations, one for the potential, one for the adaptation variable. Here are the parameters that we used. I will not explain all of them because of my voice, but okay, membrane capacitance, refractory time, threshold potential, reset potential after spike, leak conductance, leak membrane potential. These parameters, A and B, are for the adaptation variable. These deltas are for on the how steep the exponential curve is after spike, and the time constants for the adaptation variable. The voltage has a synaptic current component that can be decomposed into the contributions of AMPA, GABA, and NMDA. AMPA and GABA is easy to calculate. We just multiply the conductance by the difference of the potential right now to the synaptic potential. And this conductance can be calculated by this differential equation. And each time there is a spike, this equation is added by the synaptic weight. So what this says is that each time there is a spike, there will be instantaneous jump in the conductance of the channel, and then it will decrease exponentially. For the NMDA current, it is a little more complicated. Firstly, because it increases fast, but not instantaneously. And then because it has also a dependence on magnesium inhibition. There is taken into account by this variable B that depends on the potential. In his paper, Dostache calculated this NMDA conductance by using two more differential equations. This differential equation S, there is an activation variable, and this other equation X, there is a auxiliary variable. He called it a auxiliary variable, but he didn't give any further definitions about it. Besides that lack of definition, there is this term here, there is the multiplication between the two variables, S and X. And after speaking with Professor Hams, we learned that this operation will be very computational costly to do nest. So we decided to alter the model a little by not using these equations and modeling the conductance of NMDA by a function Z that multiplies by the maximum NMDA conductance, that is the weight of the synapse. And this function Z is nothing more than a subtraction, a difference of exponentials that has two time constants. One very large decay constant, because the conductance decays slowly, and one very small rise constant. Other than that, we keep it the magnesium inhibition dependence by this equation. And the concentration that we used was 1 millimolar to barominic biological conditions. So here I did some validation in the model. Here occurs a spike in the presynaptic cell at around 10 milliseconds that produces this alteration in the membrane voltage that you're seeing there. And there is like both AMPA and MDA channels respond to the synapse. So here in AMPA, you are seeing what I said, an instantaneous jump to the weight that I set, that was 5 nanosiemens, followed by exponential decay. And here in the current is the same behavior, but the opposite, because there are positive ions entering the cell. To NMDA, I showed here in two conditions. There, in the 1 millimolar condition, you see a fast increase, but not instantaneous. and a slow decrease. When I double the magnesium concentration, you see the same behavior in the conductance. But here in the current, you see here that it has half of the impact. There, it reaches -2. Here, it reaches -1. So this way, we see that the model is behaving in the way that we intended, despite the simplifications that we made.
Okay, so yeah, instead of doing just like random conductivity, we wanted to kind of check out how this behaved with some more structure. Now I was thinking of just kind of like a 2-layer model, whatever. I'm not claiming, biological realism with this one, but we have this first layer here of an excitatory and inhibitory population, both receiving the external driver stimulus, and then the second layer that doesn't receive any of that. The excitatory cells are connected to both populations of the other layer, and then the inhibitory cells are just connected to the excitatory cells. There's recurrent connections in all the populations, so that's just kind of what we ran with. Yeah, and I think this is going to move fast. I think we're low on time, but so I ran like 3 major conditions, but these just kind of look the best for demonstrating what we did. The top, we have baseline conditions. On the left is what we did. And then on the right is actually data from Defects' paper. Now you can see more just kind of like sporadic random firing in Defects' model, but I think that's because all cells have the same probability of receiving excitation. And then mine on the left, the green is the second layer of excitatory, and then the orange is the second layer of inhibitory. The other two are, I'm realizing now that's not awesome organization, but yeah. And so when we actually decrease the NMDA conductance preferentially on inhibitory interneurons, you get a lot more synchronous behavior, and then a lot more behavior out of the second layer. So kind of what you see here in Defects' paper, where they're kind of just really repeatedly firing at the same time, almost too much to where it looks weird. There's very little noise. Now, this is, I want to say I spent a lot of time doing this, but actually I spent a lot of time tuning my model because it wasn't working out of the box, unfortunately. But we used the membrane voltage, kind of the changes as a proxy for synaptic currents, and we're able to make the LFP out of that. So I put the cells in space and made my little electrode. And so in my 3 conditions, this is filtered for gamma, so that's why it looks like nothing's happening in the baseline condition. So you see just kind of minimal gamma activity in the baseline, and then when we drop the NMDA conductance 50%, a little bit more, and then at 0% on the inhibitory interneurons, it goes kind of crazy. And then this right here, I noticed in some of the rasters, it's when the excitation comes in, I think it's all a lot, and it's just like, starting the car out of nothing. And so the inhibitory cells get a lot of excitation as well, and then it kind of mellows out and goes into this more calm equilibrium state, I guess. So from there, I did a Fourier transform into the frequency domain to compare directly to their results. And it's not quite the same, but you see the trend. And they use arbitrary units. I followed their code to make these plots, or at least generate the data. But they kind of lost me, and frankly, it didn't work too well when I tried to use the last bit of their code. And so I don't really know what these arbitrary units correspond to exactly. But what you can see is the trend between the conditions. And it does follow what they have. And they took a lot more data points and a lot of different NMDA values. So onto the conclusions. Oh, wow, okay. I sped through faster than I thought. So, I mean, I guess like the main conclusion, if any, is that we were able to use a simplified equation and a bit more of a complex architecture within the network to kind of, get similar increases in gamma when you reduce the NMDA conductance in the interneurons of that network. And you get an increase in gamma band power in the frequency domain as well. So to further kind of expand this model, I think what would be nice, I mean, as the, I guess, biologist of the bunch, is just kind of add more biological realism and use this I don't know how to say their name, Potchins and Diesman, yeah, that model of the cortical column. The connectivity is definitely much more complicated, and they get thalamocortical input to 2 different layers. So I think that'd be interesting to kind of just apply the same thing and add a little bit more, as if we had more time. I would probably try and do something like this, but just given the month, I wanted to get a result. And then, in addition to the more biologically relevant connectivity, I think what would be interesting is use our exact same model, maybe try and do it in Brian 2 or something like that that could handle doing all the math that Defects put into his, and see if we could get the same results with the the more planned connectivity. Yeah, and then obviously taking those equations and then putting it into something like this would be cool too. So yeah, that is about it. There's references to if you want to see if I'm lying, but yeah, that's all.
Some of the models that work with the AdEx models in that configuration, they'll have actually very strong adaptation currents. You'll see large excursions in the membrane potential. Do you see that there?
Yeah, definitely.
Large, very strong, like negative 100 millivolts.
Yeah. Yeah, when I was looking at because I didn't know that, I've never used that cell model before, so I looked at the voltage traces and I actually spent that was like half of my day. I was trying to figure out what went wrong exactly. But yeah, there was a bit of that. Yeah.
Some of those values may obscure some of the effect that you have.
Yeah, yeah. So, I mean, I thought about these things, but I wasn't, I don't, yeah, no, definitely. I just, I kind of followed the code that, Defects was nice enough to send us his code directly because it wasn't on ModelDB, unfortunately. So I just kind of followed what he did for that. Yeah.
I get a question on your choice to use multiple layers.
Mhm.
So what is the, now you took it 2, you even would like to go to 6. Okay, so
Yeah.
what is the rationale for it? Do you have some specific reason for willing a layers capture?
Yeah, I mean, from just what I know about, yeah. Yeah, I mean, from what I know about these impaired gamma oscillations, I look at them particularly in auditory cortex, just kind of like in my work. And that's very layered structure with not exact, but pretty well stereotyped connectivity. Now, I mean, I came up with this idea for the project before you made the whole point that structure is not crazy important. But yeah, I think just, layering it up to what I've seen and what anatomists have seen in the past might just either confirm the results even further or, find something interesting and new.
But actually, it's a very good idea. So if you want to continue this project, as a matter of fact, you need the layers. That's important because some layers are feed-forward senders.
Yeah.
It's a layer to the receiver.
Yeah.
If you do some realistic connectivity, it may become interesting also for interpretations, for instance, for why gamma is linked to lack of sensory integration or hallucinatory phenomena. So this could become very interesting.
Yeah. I think it'd be interesting too to, I mean, this is like way extension, but to see how it kind of behaves in like 2 columns interacting. I mean, there's this idea that gamma is important for communication between areas, so that'd be even further, maybe a long time down the line if I were to actually continue working on this.
Why did you learn?
Do you want to, no? I mean, I can keep going, but.
You mean in
Yeah, okay. Yeah, I mean, I learned a lot about just like tinkering with the model and getting frustrated with it and how you just kind of have to keep playing around until you find something that works. And then making sure, the solution you found makes sense also. And then kind of just figuring out, nest and calculating LFP from these kind of things. I've used, things in my models to calculate LFP before, but I never actually wrote the code to do it. I just, ran a function. So it's nice to, kind of see how the sausage gets made, I guess. Yeah, thanks.
Hello. Okay. And well, good morning, everyone. And well, we'll talk about our project that we have been developing here, and it's called like Defects and Abnormal Plasticity in the influence of it in our random network. We use like the program and everything. We simulate everything using NetPyNE, and well, in some different statistics too to analyze like synchronization and etc. And well, how this presentation is projected is like separated. First, we'll talk a little bit like why we did this, why we studied the different abnormal plasticity, why it's important to understand it. And also then talk a little bit about the AdEx model that the Pedro and Scott talked about earlier, and a little bit about the SDP rule that we have used in this, and some statistical tool that we have used, the results, and well, the conclusions. And well, why is interesting to study this in first place? Because in like prenatal children, it's very common, like for them to have, like it's not common, like it's the usual, that they have many plasticity and also many synapses that occurs on them. And because of that, some has some defects that they are very low or even have a high plasticity. And due to it, they occur to have some kind of synchronization and spontaneous epilepsy. So it's interesting to understand like how this kind of plasticity influence in the network, how we can study it, modeling, and see other types of treatments and etc.
Our work is a random network with statutory and inhibitory neurons, where each single neuron was modeled by AdEx model. AdEx model is an improvement of leaf models that have AdEx model have two equations, one for membrane rotation with a exponential term, and other for the adaptive currents. So we have to put the spikes by hand. If the V reach the peak, we reset the rotation and increase the current. So AdEx model can reproduce spiking patterns like this. So we can play around with the parameters and reproduce like this initial bursting pattern in blue. So our model initially has two populations of neurons, one is statutory and other inhibitory. The statutory neurons make STDP connections both with statutory and inhibitory neurons. STDP synapses have weight that can be increased or decreased depending of the interspike interval between neurons. If the pre-synaptic neurons spike before the post-synaptic neurons, we have potentiation, so the weight increase in the synapse. We can have a maximal or minimal value of increase or decrease if this difference is a small number. So we can analyze these results with some mathematical tools that Diogo will talk about now.
And well, the model of the SDP that we have used, it is on the reference, like the paper. It doesn't model like the real biological thing, more like the phenomenological things that happen in plasticity, focus on like the NMDA receptors. And well, so what did we use here to check, to analyze our network? So firstly, we use the CV, the coefficient of variation. Here is interesting because this, in many integrating fire neurons, we can use this kind of statistical tool to analyze if the neuron like is on spiking or in bursting. And for like most of models, we have a very specific threshold for its CV. If the CV is below 0.5 in the AdEx model, we have spike in the network, or like in just one neuron. And if it is above 0.5, we have bursting. Of course, we don't know what kind of spike, if it's adaptive, tonic, or the bursting is regular or irregular. But well, we can check what kind of pattern do we have. And calculated, we have like standard deviation and the mean interspike interval here. And to calculate this, we just need to check like what is the distance, like, what is like the time, the difference of the two spikes that happen in the neuron. And well, and also to check now the synchronization, we use the Kuramoto order parameter that is necessary to use those three equations here. This is the Kuramoto along the time that we can check it. And to calculate it, we need the phase to calculate here. Just one important thing here is just a little bit about the math here, but this M that we have here is not a kind of power. It just means the mth firing of the neuron. So like if M is equal to, is like the time of the second firing of that neuron. It's not like T power 2 is like to square. And then we can calculate the mean Kuramoto order, like to check or if the network was synchronized in a kind of timing interval here. And well, so a bit about the results. So the network was composed of 100 neurons, where firstly, like the first step that we take was just two populations. One excitatory, another inhibitory, where 80% of the neurons were excitatory and 20% was inhibitory. And then we have the connections between each population and well. And then what did we do? We want to understand how the potentiation factor affect the network. How, like, if we increase it, what would happen? Firstly, what we have done? We took just a small potentiation factor, very small one, to see what was happening. When it's low, we have some kind of little synchronization of some neurons here that we can see on the traces here. But in general, they are desynchronized. They are like firing in a very specific time being that we can see here. Some are like outside of it. But in general, they are desynchronized. Like, because here, like the Kuramoto order, it's about 0.4. And to be synchronized, the Kuramoto needs to be like close to 1. But usually we can define a kind of threshold to see if synchronized or not. Usually people take like 0.75, 80, something like that, to see if it's synchronized, because it's very hard to reach 1 specifically. And well, and what happens if we increase it now? When you got like for potentiation for 0.06, it starts synchronization here, but not just synchronization of spikes, but synchronization of bursting. And why this is bad? Because it's totally related with epileptic seizures that can happen. So when we increase it potentiation, we can get to synchronization of bursting. But the thing is, here, all the neurons that have the STDP rule, the excitatory neurons, have this potentiation. And the interesting thing is like, why? Like, is necessary all the neurons to have this abnormal or just a few of them? And well, before that, how does the weight, like, what is the connection matrix here? What is the Jensen matrix, like, when the weight is too strong? Well, this is the initial condition for each case that we worked on. And, like, the excitatory is more or less 10 minus 3 nanosiemens. And then the inhibitory are double of it. And what happens, like, after 8 seconds? One important thing here is to see this. Here we have like 10 minus 6, but once we have a very high potentiation and increase the time, like the 8 seconds after, it becomes like this. It's way colorful than before. And the power increases like by 10. And this is interesting because we have some nodes here that are it's hard to see here, but they are red. They have a lot of weight here in their connections. And why this is interesting? Because many papers have shown that when we have networks of AdEx model or other kind of leak integrated fires or integrating fire neurons, the coupling plays a great role to achieve like synchronization. And well, since we have these neurons that have a lot of this, so they can play a great role here. And well, and then we try to see what were like when it starts synchronized here. Those dotted line in the CV, it means the threshold for the transition of spike to burst, and here the transition from asynchronous to synchronous behavior. And we see as we increase it, the potentiation, it starts to transition here, and also we have like more synchronization. But interesting thing that we can have here, more or less, bursting, but not synchronized. But as we increase, we get more synchronization. And now the second step that we had, now we have 3 kind of populations. The excitatory neurons with regular STDP, that we use this kind of potentiation, this value, and a ESTDP, that would be excessive STDP that we call here. When we increase it for that value that we assume here, that we have a lot of synchronization and bursting, we want to see like how many neurons in the network were necessary to achieve the synchronization of bursting. And firstly, what was interesting, when we have just 5% of the neurons with this ESTDP, what happened? We don't have spiking anymore. It goes for bursting right in the beginning. Like we don't need many neurons with excessive plasticity to reach bursting. However, their synchronization is kind of low yet. We need to achieve a synchronization of bursting way more excessive neurons. So some neurons, more neurons need to be like with this defect in the network. And well, the conclusions. The potentiation factor is like can trigger synchronization, the spike to burst transition, and also it plays a great role like when we have just a few of them. Like it's not necessary that all the neurons have this problem, have this high plasticity. Just a few of them are necessary to achieve the synchronization them. And well, and also we want to check also something, what would happen if the depression factor were too great? Because there are other diseases that are related to the lack of plasticity. One of those is the syndrome X fragile, that is a cognitive syndrome, and it's totally related with the lack of plasticity in the brain. So it would be a nice thing to test it also, and to, of course, go to more biological network, using maybe a more biological model to check it. And well, some acknowledgements for the event, the universities that we studied, and the groups, and some reference here that we have used. And well, thanks for the attention.
So thank you for this very nice presentation. My question is about the way that you were measuring synchronization. So the chromotor parameter, you're performing an average, right?
Yeah.
And if the chromotor parameter is equal 1, we know that it's perfect phase synchronized. But when the chromotor parameter is lower than that, in between 0 and 1, how did you check? Because a chromotor parameter of 0.5 can mean different things. It could be some kind of disorganization, or you can have some kind of anti-phase behavior, or some kind of diagonal structure, for instance. Did you guys check for this kind of thing in the raster plots?
Yes, was totally like just by looking, we test many of them to see if it was synchronized, if it were not. Then like after many of those raster plots, I'm just showing like 2, 3 here because we're way many that we tried there. And usually like for 0.75, they look very synchronized already. And we check like some neurons, some other combinations here that we took to see if they were really synchronized. And this was like a good parameter that we chose.
So I was interested in how did you implement the SVP specifically? What are the bounds on the synaptic weights?
Well, we chose, I took this, like the maximum weight that we used there was around 0.3 nanosiemens, and we choose this because many works have took this as a boundary, because once we get more than 0.2, it starts to have a lot of bursting and synchronization already. So we took just like let's set it very high so we can see different things there.
And how long did you run the simulations for with the plasticity?
Well, firstly, we're testing a lot with like 3 seconds, but it seemed too short. Then we increased it like to 8, but we tried 8 seconds, but we tried with more, but took way much time because like the computational power that we were using were like very low. So we need to reduce for 8 seconds.
And how do you think so the last result there, how do you think that would change? So, real brain networks are, over hours or days, and how is that going to shape this result?
That is a good question, and I have no idea so far. Yeah, that is a good thing, like to even try to work with a more biological network here, because it like shows that like it's not all neurons in the network that need to have like this kind of potential, just a few of them, but how more connections, more neurons would affect it, would reduce it, would increase. So this is interesting to think about. Yes, probably, because here we use like was a random network, and the probability of connection that we used was 0.45, if I'm not wrong. So the number of connections were about like the mean I think was like 60, 90, no, 60, 15 per neuron, where it was a small number. So increasing it would like probably refine way more the results.
That was super cool thing in your work, that the difference between when you just have all the neurons strongly differentiated and just a few of them, the symphony could be the same, but the CD is systematically smaller. I didn't get it to my dream. Do you have any interpretation?
Well, that this like the change of the CV that we got here is mostly, at least that we think about, is because some neurons, since we have this kind of big potentiation here, they starting to get way more, like the weight start increasing way more than before, and then we get to that situation as before that we have some neurons with a lot of weight here, and they influence a lot of the bursting.
How you get it, but dynamically these states are different. Because in one case you have a synchronization of that you have network burst, but you have
Okay.
neuron burst. The CD is about the neuron burst. While here you have synchronized neuron burst. In the other case you have synchronized network, but the neurons are still constant. That's interesting. That may be something you could go in, because it's the difference. What if the individual neurons are bursting, or if the neurons are still relatively regular, but the network is bursting? And I think that there is something to study here.
And then like I didn't put here, but was very like curious in this case here, that when we were checking all the data, everything that we got around here, when we have the potentiation like 0.03, more or less, this point here, we have like synchronization of burst, synchronization of spike at the same time. You can see in the raster, just it's very interesting.
The neuron is bursting.
Yes.
But on the other side, you can have synchrony as at the end of this line, but the CD is still around. So I think it's a very different regime.
Mhm.
It's something that you can keep them. But that's
Oh,
nice.
oh, that's good. Thank you. And
So we were with question in the ASCON.
and well, so firstly, we learn a lot how to use the NetPyNE. Like, firstly, I was not like used to work with it. Like, NetPyNE was kind of hard to use, even the mods to understand them. But Valerie and Fernando helped a lot to understand the mods for the STDP, for the AdEx, and then to play with the parameters took a lot of time. It was a very simple network, very simple model, but the parameters of the STDP, was very bad to work with it. Like need to change because of the paper that we took it. They were using parameters for like very biological thing, morphological thing. So we need to like to change the parameters, change like the order of parameters to work with. So it was nice to see how it would work and the NetPyNE and everything.
Control what? And then something. Okay. So fortunately, my colleague, my partner is not here anymore to help me show you guys our lack of results here because it didn't work how we expected. So main idea was to study the effects of dendritic tree morphology on information processing at cell level, comparing pyramidal cells and Purkinje cells, using this idea that
you can find usually Purkinje cells mainly in the cerebral cortex, have totally different functions in the brain than the cerebral cortex where you find pyramidal cells, right? And the morphology is totally different, right? But we don't reach the point where we can really say something about the information processing, and we also didn't really explore the Purkinje cell yet. An overview here. So this was the idea. And we had the idea first to try to build artificial cells that capture some of the morphology between these kind of cells, like to explore this in a more controlled way. So have some parameters that allow us to build dendritic trees that are more close related to Purkinje or to pyramidal cells, and explore and make simulations with really simple mechanisms like HH in soma and passive in dendritic tree. And then when we finished this part, our idea was, well, to use biophysically realistic models for Purkinje and pyramidal cells and do the same work there in a real case scenario, right? And how we will measure information processing. Our idea was to use partial information decomposition. Is go ahead, like a little bit more detail about how information is flowing in the system. Is like an extension of just measuring mutual information between inputs, variables, and a target, for example. In this case, what this framework allows us to measure is not just how much information you get from the inputs about a target, in the case of soma, but also how this information is really distributed in some sense. Like you can measure how redundant are your information between inputs, for example. It's like I measure any of the inputs and I get exactly the same information about the target, the soma. Or in the other cases, you can find structures where this information are synergistic. So what means that you need every information about every input to have a reduction of your uncertainty about the state of the target, in this case, the soma. So it's a really nice idea, framework, to try to get more information about what is happening in a system, any complex or any system at all, right? It's general. There is some works where we will find this kind of approach. In this case, they try to measure synergy between apical dendritic part and the basal part. Can I show you here? So in this case, what they found is like in pyramidal cells, the most of the information is unique for the basal part. So it's like the basal dendritic tree is driving the soma. The output in the soma is kind of obvious, like it's close to the soma. And the dendritic part, the apical part, is just a kind of context. So there is synergy between them, but the most part of the information is coming from here. It's driving, it's the basal part that are driving the soma. And when you inhibit the apical part, you will have this diminishing the synergy between structures, and everything that will happen in the soma is mainly driven by the basal part. This is the case of pyramidal cells. In this work, they didn't explore other kind of cells, right? Just pyramidal cells. So our main idea was, okay, let's also do experiments with a real detailed model for pyramidal and the Purkinje cells. So we wanted to import some models with all the biophysical details, channels, everything for a pyramidal cell, select different regions of the dendritic tree, introduce inputs there, introduce background noise in all the tree, like uniformly, make simulations, generate time series, binarize the time series, put a threshold there, obtain Boolean time series is easy to analyze, like just zeros or ones, active, not active, right? And apply partial information decomposition. Seems simple, like the work seems simple at the beginning, a priori, but we had a lot of problems. First problem is how we would compare these two types of dendritic trees. They are really different. The scales are different. Like a Purkinje cell is really small in comparison to this. So an input that you put here will be really close to the soma in comparison that input you put any place in other. So how we would compare this? This is the problem. That's why we thought, no, let's start by constructing a simplified dendritic tree to capture some of morphology aspects and explore that first, right? And to make this kind of realistic, our idea was to first measure some metrics of the dendritic trees. For example, estimate the branching probability without respect to the distance to the soma. And we did that. We tried to first obtain this distribution probability for both types of cells and use that to generate a artificial dendritic tree. Problem. 3 days ago, I find a bug in my code, and these probabilities are wrong. So all the work was just a failure. So we said, okay, let's do it with a constant branching probability and start with the most simple case, right? So we started doing that. Define it's a probability of non-branching in some sense. Build some different trees like this, this, and this. Obtain time series and analyze the results of the time series to try to extract the difference between redundancy, unique information, and synergy with these different cells, right? Problem again, we don't understand what is happening here. Look at this case. We have different points of inputs, the red dots, but we are measuring the memory potential. I was seeing exactly the same. So probably this is another bug in our code that we found yesterday. So we just say, oh, yes, I have no time to do this properly, right? So we don't even try to analyze the data here because we cannot explain what is happening still. So I'm sorry, guys. And then, okay, let's desperate measurements, desperate measures, right? Let's go to see what's happening with the realistic cell. So we did it. Also yesterday, we can manage to really start to running properly these simulations. So we have, for example, in this case, inputs in the basal dendritic part. This is the inputs, Boolean. Just activating synapses in these regions. We have noise, like 50 random synapses across the dendritic tree, excitatory and inhibitory, with some rate of, it's a possum process, right? Some rate of firing. And we measure what is happening in the soma. But we didn't have time to do really long simulations. We need a lot of data for extract properly the distribution's probability, to measure the entropies, and to apply PID. So with the data we have, we just apply our code, just show that we can do it. We have the software like ready to go now, but we hadn't time to really do the simulation properly. But they have other problems here is the binding of the time series will have an impact on the measurements, right? The time delay between, because you have inputs and you have distance, the pulse will travel some distance to reach the soma. So probably we need to explore also the delay between both variables when we measure the information. So it will be the time delay mutual information, what we will need to explore. So we need to explore what is the impact of the delay also between the sources and input. And here you have the factor distance, right? And we still need to think really how we will make these results comparable. Like the dendritic tree are really different. So we'll have a lot of questions here we need to solve before we really have something to show to you. Another thing we are thinking is our background noise is totally decorrelated. But probably there would be, like, if we want to have some real simulations, probably there will be some correlations in the background, like between other synapses, right? And it's interesting to explore how this could impact our measurements too. And also compare active spiking, like active dendritic trees and passive ones, to see the difference between not just the impact of morphology, but also the different ion channels, the different receptors. We have the density of receptors and other things. So that, yeah, we tried. We did our best, but it was not enough. And we will keep working with this because we still believe that this is a good idea. It's a good idea to explore. So, yeah, so that. Thank you. I never work with detailed models. So now I see it's a nightmare because a lot of parameters, we don't even know how to start to thinking in the problem. It's a lot of it's just this time you have the diameter of the dendritic, the sections. Which of those are most important to begin with? It's complicated. In my life, yes, because yeah, I get it. Yeah, totally. Yeah. Yeah. It take 1 month, but yes. Yeah. Okay, thank you. Yeah. We still have I think, a good hypothesis, but it's the only thing we have in this that moment, right? We need to test it really. We don't have any justification for the constant probability branching. We're just a desperate measure to have something, because we wanted real distributions of the dendritic tree, and we have to measure by ourselves. There is there's a lot of literature about it. We just could copy from some distributions or think in a way to justify a distribution there. But we wanted to measure in real dendritic trees and extract by ourselves the distribution, right? Then when we realized that I had a bug and my distributions were wrong, we just, no, let's put a constant and make something just to generate trees. It's not justified at all. We don't even know if even the literature. I didn't have really time to sit and make a lot of literature review of everything. Yeah, we changed idea a lot during the move. Yeah. Oh, the Purkinje, for example. Mm-hmm. Yeah. 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 Mm-hmm. Yeah, we are planning to do like hundreds of simulations with inputs randomly, or thousands. Yeah. The minimum, right? Yeah, we knew that. Yeah. I answered already everything. Yeah, on this. Okay, thank you.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, the really the best way to learn.
Good morning, everybody. My name is Peter.
No, wait until
Okay. Sorry.
So let's be.
All right. Perfect. No, it's. Okay. Good morning again, everybody. So today we'll be sharing a model with you guys today that we built that takes astrocytes, this type of glial cell, and calcium dynamics as an integrator of neuronal activity and how it might influence downstream neuron activity. And so it's not very well understood how astrocytes respond to neural activity, but it has been shown that you can have a pretty wide range of integration ability from, astrocytes responding to afferent neurons that are just sort of passing through an area. You'll get, different types of calcium activity waves in the astrocyte. They also can respond differently to local neurons, and then you can also get a different type of response from when they're integrating both local and afferent activity. And the output of these various calcium waves in astrocytes or different types of spiking, whether it be, you can get these spreading activities through, pretty large ranges of astrocytes connected by gap junctions. Or you can also just get sort of microdomain activities, and decoding what these different calcium waves mean in astrocytes is a big important task in the field right now, and understanding how astrocytes might affect neuronal activity. And so this is sort of the main principle that we wanted to model today because it's pretty poorly understood. And so we fortunately found this paper that came out in 2019. It's really cool using zebrafish larvae, which are transparent, so they make for nice in vivo imaging studies. And what they found is that radial astrocytes, which are really typically found just in the developing nervous system of vertebrates, where they actually form these projections and actually can almost synapse, I'm hesitant to use that word, but really form these projections and can release gliotransmitters onto neurons. And so there's these 4 principal cell types that we'll talk to you guys about today that we first set out to model. And so what this paper was demonstrating is that astrocytes, or these radial astrocytes, are integrating both visual information and motor information to detect when a fish is failing to keep up with a current. And so this mismatch of visual and motor information is then fired through these nemoneurons, or norepinephrine neurons of the medulla oblongata, and these then cause these astrocytes to spike and then release gliotransmitters onto these GABAergic neurons, which then silence motor neurons and therefore inhibit the fish's swimming. And one cell type that we're not showing in this graph that they then had to work out was these astrocytes are not only taking information from these nemoneurons, but also the local circuitry. So both components are important.
So in order to replicate this circuit the paper was talking about, we first developed a computational model of an astrocyte with a GABAergic neuron. Here we have the astrocyte model we used to simulate the circuit. Actually, we use here a compartmental astrocyte model. So each compartment in this astrocyte was simulated like this. So we simulate the currents through the endoplasmic reticulum, through the cytosol, and the extracellular space. And to model these currents, we used these both equations. I won't describe the details of these equations. If you wish, we can just consult this preprint. But anyway, with this coupled equation, you can simulate the calcium signal in astrocyte, or replicate this type of response in astrocyte. And we also model the glutamatergic receptor, which accounts for the input arriving from this local circuit, and also the noradrenergic receptors for the NEMO input, for the NEMO spikes. Here we have the morphology we used to simulate in this project. We used a morphology composed by 16 compartments. One is spherical, representing the soma, or the somatic compartment, and with 5 processes. Each process was further subdivided into 3 subcompartments. The local input arrived just this tip of each astrocyte process. And here we have like the morphological characteristics of the astrocyte model we used. So the radii and the length. So with this model, we aim to test 2 hypotheses, or aim to test 2 questions. The first one is about the neuromodulatory input, if whether this is required to see or to detect the calcium signal in the astrocyte. And the second one was to test if the proposed mechanism for generating the GABAergic spike in the fish paper proposed is indeed sufficient to reproduce or to make the GABAergic neuron active. So here we have the first result we got from our simulations. In this case, to test the first hypothesis, to address the first hypothesis, we simulated the astrocyte model in 3 conditions. One with the local circuit input and the NEMO spike. The second condition with just the local circuit input. And the third condition with just the NEMO spike. Here in each row, we have the time series, the calcium time series for each compartment in one of the sub-processes. So the blue for the somatic compartment, the red, green, and yellow for the process compartments. In the first case, in which we have both inputs, the local circuit and the NEMO spike, we detected indeed a calcium spike. So with both inputs, we see this astrocyte active. But if we apply just the local circuit, the local input to the astrocyte, we got like this more noisy response. We lost the time precision we are seeing here with both inputs. And also just with the NEMO spike, we didn't see any response at all. So these results show us that the astrocyte is indeed integrating this local input, arriving at the sub-process or the tip of the process, with the NEMO spike. The NEMO spike actually somewhat gating this response, so it's integrating all the response the astrocyte is receiving from the local circuit, and then create a spike in this calcium response. Since here we lost the time precision for the calcium signal, we tried to explore it a bit more and conduct a second test in which we vary the time of the NEMO spike compared to this local circuit input. So we varied it at the end of the local circuit input, we applied the NEMO spike 35 seconds before the end of the local circuit input, or the end of the local circuit input, and 35 seconds after the end of the local circuit input. And we test or measure the latency, or the time we had to wait until we detect the calcium spike. With this test, we got this type of curve, which resembles the one Marco Pérez showed us for the case of like psychophysics. And we found this interesting because it's showing us that the astrocyte has like a optimal range for response, or a time range for in which it has more sensitivity to the NEMO spike compared to this local circuit input.
Okay, so part of our model was a little bit unusual, we'll admit, and so there's really not much evidence for this biologically, and so I'll go through essentially what here we're modeling, which was that the proposed gliotransmitter released after this astrocyte calcium spike is potassium. So they're actually directing potassium release onto these neurons to generate spontaneous firing. And again, there's not much precedent for this, and so we just, the reason we thought it plausible is because many of the other papers demonstrating gliotransmission in terms of the release of glutamate or ADP or D-serine or other types of canonical gliotransmitters happen on the time scale of seconds or minutes. And so they were proposing here that this fast time scale is actually due to the release of potassium, which in theory would then raise the extracellular potassium concentration, then raising the reversal potential for potassium. And so we wanted to know if we then could, just with no current input, if we could generate spontaneous firing in this GABAergic neuron by raising the extracellular potassium concentrations. And so we started, our default value in NetPyNE was around -80, where of course the neuron is silent. There's no spiking at that reversal potential with no input, and tested different increasing reversal potentials for potassium. And when we got to around in between -62 and -61, we started seeing spontaneous firing with no input. And again, this is what we modeled as concurrent with the astrocyte spike time. And so this is great. Our model was able to, and this makes sense, of course, that a neuron, if you raise enough potassium, that you can get this type of spontaneous firing. But we, again, this not being something that's really well studied biologically or computationally, is if this level of reversal potential for potassium, or this extracellular potassium concentration, is physiological, if that could really even be possible. And really the same paper that demonstrated that these glial cells can release directly, because we've known that potassium is buffered by glial cells, but this sort of discrete and precise release of potassium is not really well understood in terms of modulating neuronal activity. And so this other study that we were getting these ideas from, that potentially glial cells could be, directing potassium release, demonstrated. So this UTP is an I believe it's an adenosine receptor agonist. So essentially this is similar to the model that so they took these glial cells in this model and were able to evoke spontaneous depolarizations here at about 13 millimolar of extracellular potassium concentration. And interestingly, to get this -60 reversal potential for potassium, that equals about 13 millimolar of potassium. So it's in line with really the one biological reference that we have. So that was exciting for us to demonstrate, some of our model mimicking reality. And so in our summary, our model was able to show that, we were able to generate this sort of at least semi-realistic model of an astrocyte integrating activity. And this is important because there's this idea of astrocytes integrating inputs of neuronal activity is really not only poorly understood biologically, but also we're lacking good computational models for it. We've demonstrated some parallels for inputs shown in vivo. This was exciting too. And of course, we have more to build upon this, but some of the basic parameters that we tested seem to be relevant to these few papers that we've been modeling. It's pretty cool that we found this broad dynamic range of astrocyte spike times, indicating that, right, like Madam mentioned in the beginning, that astrocytes really, they don't just have this one type of response, but depending on the types of stimulations that they get, they can respond differently, and that needs to be worked out on what exactly that means. And lastly, that this is important that this potassium released by astrocytes, we showed that this matches what is demonstrated in vivo, but we need better models both in vivo, in other systems, but also to model that. And so we think this model can help improve that understanding.
And just to finish, as our works, we have also some limitations in our project, in our model. The first one is that the astrocyte lacks complex or realistic morphology. We are using a pretty simple morphological model for the astrocyte, like 16 compartment, star-shaped. And we know that astrocyte has a complex morphology. So this is the first limitation of our work. The second, we are using somewhat simple upstream neuronal input to the astrocyte. We simulated this local circuit input as Poisson spikes, not exactly really a neuron, but anyway, it's a second limitation. Third, we have no feedback from the downstream GABAergic neuron. So this neuron also could influence astrocyte response, but we, in this case, did not simulate that. And finally, we are assuming that the glial transmitter release happens in the millisecond scale. In general, astrocyte has a slow time scale response. in this case, as the author proposed, actually, in the fish paper, so we take here also that this gliotransmitter release happens in the millisecond scale after the astrocyte triggers the calcium signal. So that's it. Thank you. No, we did that. I mean, it somewhat captures a simplified version of astrocytes since they have this star-shaped. But yeah.
And then a second comment. So this would be perfect to see stuff that was in the previous project because this idea that you have the single the Nemo spike is not enough or the background spike is not enough, but two together are enough. That's a perfect example of XOR and XOR gate. Is the contrary, is the N, is the non-not, is the NOR.
Yeah.
With the snore, but it's the same. It is also synergistic.
You use the model that you already had before coming to LASCON?
Not exactly. Here, I didn't have this model, since here is working with the simplified version of this model. So we did to build like this to get for the project.
So yeah, what is the novelties that you incorporated into the model, and again, what did you learn by doing this?
It's interesting because I didn't expect how cumbersome could be to simulate astrocyte within a neural network. So we needed to like manage how to simulate the astrocyte within this the neural or the output, because at first we are trying to simulate both together, I mean in the same like code, but the simulation was take forever to run, like 4 hours, 5 hours to run just one simulation. So we tried to like somewhat decouple both models in order to simulate the astrocyte and use the astrocyte as input for the neural network. And I wasn't expecting to have like this trouble in simulate both together. Since the astrocyte has a time scale of seconds and the neurons has a time scale of milliseconds, it can get you can get in trouble simulate both together. So we needed to do something different to be able to simulate these here to present for the project.
Can I just add to that? Well, because I'm a biologist, and so what I learned was I simulated this GABAergic neuron in NetPyNE, and we had to figure out how to input, your customized reversal potentials, and so all that was new to me is, taking from what was known, but that part was, so I mean, it was the simplistic part relatively on the coding, but yeah, it was a lot to learn for me.
So I may have missed this, and so you're changing the reverse potential of potassium in that last result there, and that's mediated by release from astrocytes and so changing the extracellular. So I wonder whether how the timescales fit in, and so that's actually not going to be instantaneous when the astrocyte releases and then changes the extracellular concentration. So how do you think about that playing in, or is it negligible, the timescale?
No, you can simulate that, but for example, we took the calcium spike timing and used this as an input to the neural network. So we simulate, like in this time, you have a release of potassium, so you increase also the reversal potential.
But
so
that's instantaneous.
Yes.
Yeah. And that might not be instantaneous.
No, perhaps not. I mean, the paper we are using as base for this work says it is in millisecond scale, so we assume that, but we are not sure. I mean,
Maybe it's fast.
yeah.
Okay, thank you.
Okay, good morning, everyone. We will present our project called thalamocortical model for post-sleep memory improvement, a reproduction project. So our talk is to abroad six parts. We will present the objective, starting with the considering. One of the main aspects of the science is the reproducibility of the results from application of methods. That means if we apply a certain method or an equivalent method, we can achieve the same results. But actually, in practice, it's not exactly like that, you know? That's because depend on who design the results or who make the method. That's why our objective in our project is reproduce the model presented by Capone and et al. The 2019, called sleep-like slow oscillations, improve visual classification through synaptic homeostasis and memory association in a thalamocortical model. The article highlights the using of deep sleepy like oscillation for memory. So the objective is the find of objective for the authors. The study provide valuable insights into the underlying mechanism involved in the neural processing during sleep. And for our point of view, reproducing and replicate the outcomes reported by the authors, hope to provide a detailed account for their model development and findings. Well, this is a network chapter. They are composed for 4 populations. 2 belong for the cortical regions. And so we have excitatory cortical population, inhibitory population pertain from the cortex, and 2 for the thalamic, the excitatory thalamic and the inhibitory thalamic. The excitatory connections of the thalamic network receive a visual stimulus. The Catalina will show about it soon. And here is this flags mentioned about the connectivity in the networks. For example, excitatory cortical population is excite the inhibitory, and vice versa. So each neuron on the network is modeling by the AdEx model. Here, a quick remark: delta V govern how faster or how slow the membrane potential reach the threshold is very important to the simulations. So as we know, VT is membrane potential, tau m is the membrane constant, C is the membrane capacitance, EL reversal potential, and delta is the threshold. Also, the synapse model of the network is modeling by the STDP synapses. And here we have 3 populations with STDP synapses, and 4 for connectivity is static synapses. So from the cortical to the cortical is STDP model. For the thalamical, aquí is CX, is cortical to thalamic is also STDP, and thalamical to cortex is also CDP. The rest of it is static synapses.
So for the classification task, what the paper proposed is training an unsupervised learning model with MNIST images and giving that to the network so the network can do this classification task. And the images used for this classification are the MNIST images. That's a database of handwritten digits used for normal training image preprocessing systems and testing in machine learning. And this dataset contains 60,000 training images and 10,000 testing images from digits from 0 to 9. So to these images, following the paper, we applied a Hog filter That's a feature descriptor used in object detection in computer science. And it's a feature descriptor that is based on the magnitude and gradient orientation of parts of an image, as you can see here. And this is an example result of the first training image in the MNIST dataset. And you can see here on the right, the original image, and here on the left, the Hog filtered image. After applying the Hog filter, we did a vectorization of this filtered image. We used cells of 14 by 14 size, and each cell provides an histogram with 9 bins. And then each of these 9 bins is transformed into a vector of 4 truth values dependent on intensity that go from ultra low to high. Finally, this creates a vector of 324 binary features. And this vector, this resulting vector, is used as input to the network through the thalamus, through the excitatory neurons in the thalamus. And each feature provides a binary input to a different thalamic cell. And again, as an overview, the objective of using this is to create an unsupervised learning model that can do this character recognition through the thalamic representation of these preprocessed images. And we'll talk about this classification and the challenges of this a bit in the end of the presentation.
So you have just seen the description of the model and the dataset with Flavio and Catalina. So now let's understand how they work together in these learning phases to produce the results. So the first phase is the training phase. In this phase, yeah, I can point. In this phase, we have a Poisson spike train of 30,000 hertz to these dynamic neurons, but only the neurons that match the element 1 in this binary vector that Catalina just described to receive the input. So we have a binary vector of the same length as the population and filled with zeros and ones. And when there is a 1 that matches the neurons in that population, they receive this spike train. We also have a Poisson spike train of 2,000 hertz to the excitatory cortical population. And this Poisson spike train goes to groups of 20 neurons, not all of the population together. And the goal of inputting this, oh yeah, sorry, I was explaining and forgot to mention the image. So for example, here in the image, we can see that only groups of 20 neurons spike together with the thalamic input. So we have the thalamic input that matches the binary vector. So for the ones we have these black strips that would be the spikes, and for the white strips would be the zeros of these vectors. And then we have this cortical, these 20 cortical neurons firing together. And why do they do this? This works as a coincidence signal to facilitate these neurons of learning these stimulus, in this case, these images. And they also input 10,000 hertz Poisson spike train to the cortical inhibitory population to prevent our previously trained neurons of firing to new images. So that's the figure of the paper in the left, and this is our first results. So we could simulate this ladder pattern of 20 group of neurons firing for each image that we input. And we can also see this in the training phase. We could also recapture these patterns matching the zeros and ones of the feature vector. Beautiful, isn't it? But in this simulation, we didn't put the inhibitory signal. And when we do so, well, we lost this pattern. We don't know why this happens because we followed what it's described in the paper. So to go to the next phase, we kept this configuration with the excitatory input to the cortical population and to the thalamic population without the inhibitory signal. So now let's go to the retrieval phase. In this retrieval phase, we only have the input to the thalamic population. And then we don't have the inputs to the excitatory cortical and the inhibitory cortical populations. So what we see here is that in the retrieval phase, we stimulate these populations, and because of the coincidence signal that was applied previously, these neurons start firing. They are learning somewhat what they saw in the training phase, but in a more generalized way. It's not so specific as you see in the training phase, but they are firing more for the types of type of input. So in this case, it would be for the zeros, for the ones, and for the twos. So this is our results. This is you have already seen this for the training phase, and this is the retrieval phase. I don't know if it's easy to spot, but the rest of plot is equal. The retrieval phase to the training phase, they only get thinner, and we don't know why. They get thinner maybe because of the activity of the inhibitory thalamic neurons. But then when we go to the cortical neurons, we don't see this letter. We don't see any activity in these populations. And since theoretically, there are connections between these populations, we have no idea what happened, and then we decided to investigate this. So as Flavio previously explained it, all the weights of the synapses are started as ones, and after the simulation, some of the weights indeed changes. So there are connections being made, there are plasticity in this network. And then we analyzed the membrane potential of these, the first 20 neurons. So we see that as for this is the training phase, this is the retrieval phase. So here they reach the peak of the membrane potential and the fire. They don't fire here as expected for the training phase, but in the retrieval phase, even though we see an oscillation, it's not enough to reach the peak and for these neurons to fire. So we weren't able to fully simulate this retrieval phase, then we couldn't go on to the sleep phase and all the phases of this project. But then we could still study the connectivity matrix before sleep. So this is a figure from the paper. And here we see that this is from the cortical population. So we see these weights increases diagonally because these are the groups of trained neurons. And then when we did this for our network, we could reproduce this. But you may spot a small difference between the other connections. Our connections are all 1, as described, that they start as 1, and here is 0 because it's the natural logarithm of 1. But here you've seen some spots, some yellow and even blue that would be below 0. And why? The yellow might happen because of some connections between other populations as expected in the retrieval phase, but these blue spots had no meaning. So we asked for the authors about some parameters because we weren't able to simulate everything just following the paper, and instead of just answering us, they gave us the code. And when they gave us the code, we saw this. Even though in the paper they say that all the initial weights are 1, what they did is a distribution of weights for this cortical population. So that's why while here everything is 1, we have these blue spots and this distribution of weights. And the last figure we could try to reproduce is the figure 3 about the neuronal activity correlation. So here, as the retrieval phase, we have these neurons that learn a specific type of input. So these different 20 groups of neurons, for example, there is a correlation in their activity for the number of zeros, this for the ones, and then for the 2. But since we couldn't simulate the retrieval phase, we only get a correlation between the 20 neurons within them. So the summary of our project is that we could kind of simulate the training phase, but with a trick, but we couldn't roll the code into the sleep phase to the lack of the reproducible results in the retrieval phase. Then, this paper is not reproducible from its method section. But we learned a lot how to use NEST, and we learned a lot how to solve problems, and that reproducing a paper is not so straightforward as one might think. And we also learned how to face the challenge that Catalina is going to talk a little bit now.
Yes, so we, as Bruna was saying, we faced some challenges during this project. The methods section was not too well described, and and for example, after we asked the authors for help and they actually sent us the code, we reviewed the code and we noticed that there was some things missing from the methods section that were included in the code. For example, in the methods section, there was no description of the ML model used. So you may be asking yourselves, where is the classification task that I talked about previously? Well, so as we couldn't go further than the retrieval phase, we created an unsupervised machine learning model, but that kind of worked as a dummy ML model, but we couldn't actually put it together with the rest of the network, so we couldn't get to that. But it was still not described in the paper itself. Also, the code contains one neuronal population not mentioned in the paper and contains a bunch of parameters that are not described in the paper as well. So we are thankful for the authors for them sending us the code and everything. But again, and we also obviously thank Professor Hans and his PhD student Jose Villamar and everybody here in LASCON for their continuous help. But yeah, it's like we feel kind of the conclusion of this is about the problems on reproducibility on models, which is, I think, very important for all of us. So it was like output neurons that kind of represented each of the class of the MNIST images. So it was 10 output neurons for the 0 to 9 MNIST images. And with that, they modeled the whole output neurons, and then they used a supervised classification model with these output models, with these output neurons. And yeah, no. So we don't know if it maybe was some left out code. Maybe they wanted to themselves look at the difference between supervised and unsupervised, but it was a surprise for us to see that.
Noise of diagonal noise is precisely what allows the system not to get totally frozen.
So we actually didn't, even though we had the code, we didn't really want to use it too much. We used it for tests, for example, when we were still configuring all the hog filter, blah, blah, blah. We used their already preprocessed images, for example, but we really didn't want to use the code because the objective of our project was to reproduce from the methods and not just replicate or rerun the code. So yeah, we didn't try that.
Sorry, I couldn't hear. Oh.
I think we have to mention is reproduce the methods from the article, from the papers is not so easy according to the scribe of the methods, I think that is very important to form the scientific mind when you work day on day,
Yes, we were like, okay, we can reproduce the paper and then even add something on our own and make a difference. And then we couldn't even get the reproduction to the end. So yeah, it's not so straightforward as it should be actually.
So apart from that, I think personally I learned how to model because I had never done it before. I had worked with machine learning, deep learning, etc., but I had never worked in like neuronal modeling. So that was a first for me, and it was very nice, and the group was very nice too. So.
Hello, everyone. My name is Gurgen Sohoyan. This is Ramiro Plus, and today we are going to tell you about our project that was devoted to the discovery of short-term plasticity in the models of somatosensory cortex. So first of all, just a kind reminder about the short-term plasticity, which is one of the mechanisms of plasticity, synaptic plasticity between neurons that has a time scale of milliseconds to several seconds. And there are a lot of diverse mechanisms, such as, for example, depression and facilitation, which describes how the postsynaptic potential decreases or increases depending on different conditions. And there are a lot of biological mechanisms that try to explain how this can be originated from the biological perspective.
In this case, for example, hello? No, in this case, for example, we can have the biophysical models. For example, we can have the deterministic mechanism and probabilistic mechanism. There are several differences between them, but in particular, for example, the deterministic mechanism could be predictable and has defined a pattern. The synapses has a defined pattern. And the probabilistic one, it has a stochastic nature of the synaptic events and has random processes. Both mechanisms could be models of depressing and facilitating synapses. For example, for the synaptic facilitation, we could use this synaptic facilitation probability function, P of t, that follow this law. For example, we have here the P0, that is the basal release probability, and EF, the amount of facilitation per potential, and tau F, the recovery time constant. This could be a possible model to synaptic facilitation.
During the last years, decades, and we heard a lot about this in LASCON, there was a great number of different models, even those that try to model the whole cortices. And one that was devoted to the modeling of somatosensory cortex was the project by the Blue. This is a large-scale model that has a lot of morphologically reconstructed cells, neurons, of different layers, of all of the 6 layers, with predefined electrophysiologic there. And what is also interesting is that this model was even reproduced by our colleague and our tutor, Fernando Borges, and the rest. and which shows that there are like a great number of research that can be done in this field. But what was the point is that in the first project, they didn't apply any mechanism of short-term plasticity inside. And in the second project, they applied the mechanism of short-term plasticity, which was deterministic. Meanwhile, it is really a great research question. Should we apply mechanism of short-term plasticity? Because some studies say that short-term plasticity do not affect the transmission of information on a large networks, and the information transmission changes only on the small scale models. But with see those who are for this idea and with red those who are against, so we decided to shift the probabilistic STP. That shift of probabilistic STP can significantly shape the neural in morphologically reconstructed parts of somatosensory cortex. And this was the main idea of our project, which we decided to do in 3 steps. Firstly, we started to model the interaction between 2 morphologically reconstructed neurons. Those were neurons inhibitory and excitatory from layer 5 of somatosensory cortex. Then we analyzed these results and went to the higher population, where we observed the interaction between 2 populations, excitatory and inhibitory. And finally, we made a model of layer 5 of somatosensory cortex and try to understand the dynamics in that layer. All of the simulations were done with morphologically reconstructed cells.
The model of 2 neurons consists, in this case, of 2 cells. We have the soma and axon for the 2 cells. We have the presynaptic potential, the presynaptic potential and the postsynaptic potential. And in particular, we study the short-term plasticity with the facilitation synapses. In particular, we measure the voltage of the soma2, and we are going to work with these 4 synapses: excitatory, inhibitory, excitatory, and inhibitory. Well, this is 2 images of several, and here we measure the voltage of the soma, or soma2 in this case. We saw the first picture of the left is the picture for the deterministic mechanism. We see there are more finding rates than the probabilistic one, and it looks like a certain periodicity. For the other hand, for the probabilistic one, we have a lower firing rate, and it looks like more stochastic process. So we measure the average of these, for example, the firing rate and inter-spike interval. In this case, is the firing rate for the 4 synapses that I told. In this case, we can observe that for the deterministic mechanism, it's like almost constant, a difference to the mechanics one for the 4 kind of synapses. And of course, we can observe that in the determining mechanism, we observe that we have a high firing rate in general. And for other hand, for the inter-spike interval, we have something similar. In this case, we can observe that for the determining mechanism, we observe a constant behavior, like the other case. And in this case, we have a inter-spike interval higher for the probabilistic one for all cases. We ran out of time to do more simulations. That's why we don't have a standard deviation of that stuff. That could be a good idea to do in the future.
Okay, then when we understood the dynamics between 2 neurons on the small level, we decided to run the populations. So we took the same neurons, an inhibitory and excitatory neuron of L5, and built a network in NetPyNE with 40 excitatory cells and 10 inhibitory cells. we had a high probability of connection between them because actually we tried to see some spiking pattern, and we needed to make it more dense since we use those morphologically reconstructed cells. And we started with basic simulations, just wanted to understand how it works. And then we realized that when we change the background simulation that comes to the excitatory cell, actually we start to observe diverse dynamics, and that's why we decided to work with this parameter particularly and understand how it can be changed, how the dynamics can be changed when we use the probabilistic and deterministic models. And what we actually presented on the first plot, so we observed that when we apply smaller frequency, deterministic and probabilistic models completely diverge in the average spiking rate. And when we start to increase the excitatory, when we start to increase the frequency that comes to excitatory cell population, they kind of start to look more similar, but still with deterministic model, we see higher number of spikes. And this was mainly those spikes are, the difference between those spikes is the difference in excitatory spikes. So like, as you can see on a histogram, mostly the percentage of blue and red is similar, but when we see the smaller hertz, this is the 1 hertz stimulation, we see a lot of red, which is like number of spikes, number of excitatory spikes. Okay, and this is how our pictures looked like. So this was this was interesting to observe the synchronization in the first plot, which is deterministic with excitatory stimulation of 1 hertz. But when we see the same stimulation for the probabilistic model, it is completely random. I mean, it does not show any synchronization. When we go into higher stimulation, the similarity between them is like start to increase. I mean, they start to look more similar. And the main take-home message for us was that if we use like the probabilistic, the deterministic model of Sherrington plasticity and observe some synchronization, when we will make it stochastic, which is actually more natural, more biologically real, right? We do not observe this we do not observe this synchronization, which kind of makes many results that we observe like vague, right? Okay. So when we decided to run the model of somatosensory cortex column, actually it is not really a column. We tried to mimic only layer 5 with a background activity from all around, and including thalamocortical affection. On the slide, you can see the number of neurons that we had here and type of neurons that we had in layer 5. So there are pretty much of them. We use cluster for this simulation, and simulations were very short. We were able to do only 500 milliseconds of simulations for the deterministic and probabilistic model. And for us, it was more important just to run it and understand how it will behave. So here I suggest you to try to find some differences. We were able to find only, to apply only visual scanning to understand some differences between these two pictures. We were running out of time, so we couldn't do it computationally. But what we see is that in probabilistic model, we observe some synchronized state during the first 50 milliseconds of the simulation, while in deterministic, we do not observe this synchronization at all. And also what was interesting is that in probabilistic model, there are higher number of small underrepresented neurons. Those are interneurons of layer 5. They start to fire when this synchronized pattern appears. And in deterministic model, we do not observe their activation. They're not present. Like this raster plot is silent there. So our conclusions here are that the result of activity with deterministic and probabilistic models are completely different. And when we apply probabilistic model, we can observe some nonlinear rule, while with deterministic model, everything is kind of predefined despite facilitation and depression, but still in postsynaptic potential spikes appearing. And what is interesting is that from shifting to from one approach to another, you can approach like increase of synchronization, decrease in synchronization. So it is at this moment pretty random. When we, so in complex system that have morphologically reconstructed neurons, this synchronization can be originated due to such stochasticity that was applied with STP probabilistic. So in the end, our perspectives are here. We would like to run a series of the same simulations for two cells analysis and for analysis of two population models, just to understand if our results are statistically significant, because at this moment we were able to do it only for small models, only once. Then we would like to study more about the parameter space of the model of somatosensory cortex and analyze the differences between those raster plots that I showed you more computationally, just to observe the average spiking rates between different cell populations that we had. And of course, then when we'll understand those dynamics, we would like to perform a modeling of full S1 column, because as I said to you in the beginning, in the Blue Brain project result, they have a cell reconstruction for many cells from different layers. So it is really possible, but you need to come to that. In the end, we really would like to say a lot of gratitudes to Fernando Borges and Valerie Bragin. They helped us a lot with the project. They showed us how to make NetPyNE, showed us how to work with the paper. So thank you to you, and thanks to everybody who is here, who was here. That's all. We are ready for your questions.
Speak a little bit more about how you compare, because you have the probabilistic and the deterministic mechanisms there, but the parameters are very important there. How do you compare these two things regarding this parameter space, for instance?
Most probably we can start to answer from the analysis between two neurons, right?
So in the case of two neurons, we set a frequency, in this case of 20 Hertz. We work with several frequencies, but we notice for a high frequency, we observe like a saturation of a lot of spikes. We don't know if it's correct. So we work with 20 Hertz, and we vary the waves, right? And the comparison in this case is how the firing rates and the inter-spike interval evolves if you increase the weight, right? Of course, like I said, we need to do more statistics. We ran out of time, but that is the main comparison idea. We almost want to vary the frequency, yeah, but in the same case, that is the same case we had a few times, so we wanted to do more, but yeah.
Yeah, okay. And with, like, for example, two population analysis, we basically did the same. So we were trying to switch the frequencies of stimulation or switch the weights of stimulation. And with two population, we just took deterministic, probabilistic. They were the same networks. The only things that is changed is the mode file that describes how the synapse will work. And we were switching from frequency to frequency, and we were measuring the average spiking rate in our populations. Yeah. Okay.
What is your interpretation of why just the first milliseconds, and maybe there is a problem here that we see, but can you learn something?
Yeah, honestly, this plot requires a thorough analysis. I mean, and all of those simulations require. I'm not sure actually. It's really was unexpected because the background stimulation is not it is even during the whole period. So initial conditions of the network of the simulator. It is just like all neurons on the resting state and nothing else. Yep. Uh-huh. Yep. So the two systems are different, which means that probably even if you see less well, if you computed something like an LFP, it would have some kind of oscillatory component down at the end and not at the two sides. I think it's a good result, but you not rely on this initial function. Yeah. That's this usually throw it. Yeah. Thank you very much. Okay, I believe like anything you see on the slide we learned. I mean, on any of those slides, because we didn't have much no experience in computational like modeling. I work with absolutely different things. So modeling neurons, trying to connect them, trying to make new network, run parallelized simulation on a cluster. Everything was learned here. Thank you.
My case, like I'm a physicist, a reductionist maybe. I learned a lot of things because I didn't know about biology, neuroscience. So in this case, I think I understand a lot of things, and mostly in computer neuroscience too. I mean, this is a new area for me because I work in with the 12 networks, but it's like more. It's other kind of models in this case. So, yes.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's
okay. So okay. So our project is the title Computational Modeling of Neuronal Membrane Potential using the Adaptive Exponential Integrated and Fire model to investigate neural networks and synchronization. So for the motivation for this work?
Our motivation was learning about epilepsy. So we know epilepsy is a neurological disease characterized by a persistent predisposition to generate epilepsy seizures. And many studies actually suggest epilepsy seizures onset or associate with an excessive neural synchronization. So we chose the AF model to study epilepsy because it includes adaptive mechanisms, making it available for studying conditions like epilepsy. We based it on Fernand's paper and Williams' paper. And we have three objectives: develop a detailed AF model, simulate neuronal synchronization, and analyze the data when changing the synapses. The methodology we using is conductance-based exponential integrated fire. So this is an equation, the model, and
so we use two populations of neurons. Each one has 100 neurons and 80% excitatory neurons and 20% inhibitory neurons. So what we so this is the parameters we use based on the paper, and we set these parameters. And first, we did the recordings for the node 1, it's the first population that it's not connected to the second population yet. And we had this noise, it's a Poisson generator. And you can see that in the raster plot, the neurons are not synchronized with this configuration. And we did the same for the second population. We connected, this is not connected yet, so we just with the noise. And the synaptic weights for the second node are different from the first node. So we want to see when we connect these nodes, what will happen. So these are the connections matrix. We use the probability, Bernoulli probability. So this is why this is distributed like this. And what we did first when we connected the two nodes was using this weight between these connections. So the node 1 it's already synchronized with these parameters, and the node 2 has a tendency to synchronize. And when we change the synaptic weight for a more stronger, and the first node continues to the synchronization, and the second synchronize more. And we tested with this synaptic weight, this already, and they look like the same, but the synaptic weights from the second node are different from the first node. So the first node is influencing this synchronization on the node 2. So as a discussion and conclusions from this work, we have this impact of connection weights on synchronization, and we partially reproduce the results from the paper that Fernando works. What I mean by partially is because the results they have, they apply a square current pulse when we didn't, and we also didn't diminish the influence of the inhibitory neurons. So it's almost we kind of reproduce the paper, and these models are important maybe for in the future study the in epilepsy and seizure studies in the future. So we want to thank you, Hans, that helped us a lot, and his PhD student, José Villamar, Fernando, who take us a lot of questions, Professor Antonio, and all the LASCON organization. Thank you. Oh, it's the two populations.
Yeah, it's not a node, it's a label of the different configurations.
Yes, the populations are different. They have the same number of neurons, but in the node 1, the synaptic weights are the one you gave us, like G and GS tutor, and the second nodes, but between the neurons in the population, not.
Yeah, two configurations.
Yes.
Yes. The node is a name for a label.
Yeah.
And what was your reason besides why you use different parameters?
Was to because the first we were trying to do just one population exactly from the paper, and then we added one more yesterday.
You find that matrix there is like 100 sources and
Yes.
200.
It's because one population has 100 and the second has 100, but they uses like 0 to 100 and 100 to 200.
Okay, there is two populations.
Yes.
You add a new population.
Yeah.
And one of the two is more excitable, let's say, than the other for something like this because of the parameter.
Yeah.
But actually, which is an interesting idea because you know that in epilepsy you have the epileptogenic zone, but you also have the propagation zone.
Yeah.
Actually, this type of model could be interesting because it's nice that you have a zone which in principle doesn't synchronize.
Yeah.
But it by itself, but it does it once it's connected to the other, so potentially can become interesting.
Yes.
We present it in this way. So it's justified, it's better justified why you change the parameters, and it becomes a new scientific question, and then it's interesting.
Yeah.
So your second population is more excitable than the first. And I think as you're changing the synaptic or the connection weight higher and higher, they both reach synchronization. And I think the second population synchronizes earlier.
No. I don't know, but I think I don't. They actually, without the noise, they synchronize. So it's like they already synchronize without with doing nothing.
Okay, so what did you learn?
So I never worked with modeling before. I work with machine learning and deep learning. So all the things I saw in the school were new to me. So for the project, I learned how to do modeling with Nest, but I really liked also the other tools like NetPy and Neuron. And I think that's it. I learned how to model.
I'm learning Nest because working in C++. So this is new for me. That's it.
Is it working? Yeah. Yeah, should stand up. So we're going to talk about a model of two specific neurons in C. elegans. One of them is a motor neuron, and one of them is a sensory neuron. So firstly, background on C. elegans. It's a nematode. It's about 1 millimeter long. It's very good because it was until recently the only organism which people had the full pattern of connections and the full pattern of neurons, which is to a good approximation constant between different C. elegans. So, and the genome is sequenced as well, so you can understand better what's going on. And this is really cool. So it goes from being an embryo to being a full-grown adult in 3 days. And actually, if you see the embryo, it hatches in like 14 hours or so. So it learns how to move and how to do whatever it does. I mean, it's a working worm in 14 hours. So it's really impressive. People have all the neurons, and we're going to work on the head. Two specific neurons, which are a sensory neuron called AWC and a motor neuron called RMD. So our objective is to build a compartmental model using the neuron simulator of these two neurons, implementing the different ion currents which they have, and to compare that to electrophysiology data which we have. So what am I going to talk about? So firstly, the morphology of these two neurons, the sensory and the motor neuron, which will have one slide each. Then I will talk about the different ionic currents which these neurons have. And then we're going to compare that to electrophysiology data and see the results. So first, response to current injection, different knockouts of different channels, which are, if you take out this channel, you see what happens. And then there is this neuron RMD, which is bistable, and that means that it has two stable membrane resting potentials. And we're going to talk about how that could be used for computation in the C. elegans. And then the impact of the presynaptic noise on those bistable resting potentials, and that the noise can make it switch. And yeah, Schmidt trigger is an electronic device which is, it remembers, it looks a lot like this RMD neuron. So the sensory neuron, here you see the worm. It's about 1 micron long, this long dendrite. It's really small as neurons go, and it helps the worm smell different chemicals and go towards them. For the membrane passive conductance and the resistivity of the medium inside the dendrite, and for the capacitance, we used values which are reasonably constant between different cells. And if you find the space constant, it's really big in comparison of the neuron. So that calls into question if you want to make a compartmental model of something which is so small in comparison of the space constant. But on the other hand, since the dendrites are so thin, the speed of propagation along with them is really slow as well. So it takes more or less 4 milliseconds for the signal from the point to get to the soma. RMD, it has the same space constant basically, and it doesn't have such a long dendrite. And it's responsible for this reflex where if you touch the C. elegans, it'll go back. So the ionic currents, there are lots of currents, so it's not important to understand them all, but like just to get a feel for what we're doing. They're all Hodgkin-Huxley currents. Some of them are voltage gated, and this is a model of one of the voltage gated potassium channels, and there are several. There's also calcium voltage gated channels, a passive sodium channel. Well, anyway, they have different channels, and the point is that there is no voltage-gated sodium channel in C. elegans, which means that there are no action potentials in the classical sense, the way you will see them in flies or in humans. Yeah, this is C. elegans.
Which one's the clicker button? Top one? So first we start off with amplifying the currents through the AWC neuron at different currents. We actually want to see what's the minimum amount of current we need to inject into our model at the soma to cause a quote-unquote action potential. We kind of find out that that's going to be in the area of around 55 to 65. We choose 60 picoamps. Now, why did we look at injecting all these different kinds of currents? We we did that so that we could try and find out what is the delay between injecting current and actually hitting the soma. In this case, we actually do the injections at the soma, but on the next slide, we do the injections at the dendrite. Again, one of the points we have using a compartmental model instead of a point process model is because we want to actually figure out if we inject at the dendrite, is there a substantial difference between injecting at the soma as in a point model? And yes, there's a 4 millisecond delay. The red line indicates when the dendrite is hit with the current, and the gray line indicates when the soma receives that current, and there's a 4 millisecond delay between receiving that current and when the current is actually applied. Now we do channel knockouts, and first we start with the voltage-gated potassium channel knockouts, and I'll admit, these ones don't really matter that much. There's not much of a big difference between knocking out these channels and the wild type. However, if you look at some of the other voltage-gated potassium channels, you'll see that there is a slight difference. Green, i.e. KIR, doesn't show that much of a difference, but KV10 shows that it decreases, or when knocked out, it increases the maximal amplitude of this quote-unquote action potential. KV7 doesn't actually increase the maximal amplitude, but it does slow down the decay of that transient. Ironically, they actually seem to go hand in hand, because although the maximal amplitude of KV10 knockout is higher, it actually reaches the plateau at around the same time as KV7. In the voltage-gated calcium channels, you'll see that the calcium CAV1 isn't really that important. It does decrease maximal amplitude, but CAV2 is extremely important. In fact, without it, it doesn't even seem like this action potential occurs. Yeah. Oh, and this is the part about bistability. We mentioned before that RMDR is a bistable neuron, and because of that, it has two states in which it can be. It has a state 1 resting potential, and it has a state 2 attenuated potential. You can call it that. We'll refer to these states as state 1 and state 2. In order to transition from state 1 to state 2, we find that we would need around 3 to 4 picoamps of current in order to cause the switch. However, if you look at the time it takes, or the current it takes to transition from state 2 to state 1, it is much higher, at least in magnitude. You need around negative 15 picoamps to actually cause that transition. We also want to focus on this bistability because those voltage thresholds at which they actually can cross from one state to the other is different, but the fact that they have different currents is unique. Doing the channel knockouts again, we find that most of the potassium channels aren't useful except for KV3, in which case, without KV3, there is a substantially slower decay of the action potential. And in the calcium, voltage-gated calcium channels, we see that they are all responsible for increasing the maximal amplitude and therefore decrease the maximal amplitude when knocked out. However, CAV2 seems to be much more responsible, and CAV3 seems to have almost no effect when it's knocked out. What does this mean? Well, if I want to actually see if bistability is possible, I need to then follow up to this by injecting them with a lot more current than we're used to. Instead of injecting them with 3.5, let's inject them with 20 picoamps. You'll see that when we inject them with 20 picoamps, that initial resistivity to changing states is almost gone. And the CAV3 knockout actually reaches a really high amplitude. However, even though it reaches that high amplitude, it does not stay at that bistable state. Without CAV3, it's impossible to switch to the S2 state. However, if you do check for CAV2, when you do inject it with enough current, it can stay at the bistable state. So the knockout for CAV2 does not affect bistability, but it does affect your maximal amplitude. In vivo, we have seen that, from papers, you can actually randomly switch between states, and this can be caused by, theoretically, a large amount of synaptic noise. We have been currently using only pulse and constant injections, so we wanted to follow up to this in silico by introducing noise in our current. Then we asked ourselves, well, if this noise is existing in C. elegans, they need to have some amount of resistance to the noise. You don't want to be switching states super often. So by applying our previous methodology and combining it with this, we apply the synaptic noise to multiple knockouts. As you can see in the calcium knockouts, yeah, if you knock out those voltage-gated calcium channels, the rate at which you can switch between states is drastically reduced. However, when you knock out potassium channels, it actually doesn't change, and arguably you could mention that it actually increases the rate at which you switch between channels. or switch states.
So this is a lot of information about different channels. So because there are no action potentials in the C. elegans, it's hard to say what is the most important part of the response and what is the least important part. But also, it works. So the worm is able to forage and find food, and so it's not completely messy. So an idea we have is that, and people have talked about this electronic component called a Schmidt trigger, which is, it has two stable voltages which it can output, and it acts essentially as an analog to digital converter, where you can convert just to two digital states. And this is really what the RMD neuron is doing. It receives input from presynaptic sensory and interneurons, which is noisy and analog, and it outputs a signal which depends essentially on whether it's on this more hyperpolarized or more depolarized state, which is what people have observed multiple times in the worm in experiment. So this is useful because since the neurons in C. elegans are so small, it tends to be a lot of noise, because you think each channel has a conductance of around 5 to 10 picosiemens, and so if you do, it's like 100 channels in the membrane. So there's a lot of noise from thermal switching. This would be useful to diminish that noise, and since it's a motor neuron, it has to have a certain stability with respect to noisy synaptic input. And this is exactly what you see in vivo and in the simulations, where if you inject too little current, you see that it doesn't switch, it just stays in the same state. And if you give enough current, it goes to the other state. If you give enough inhibition, after that, it returns to the state it was previously. Yeah. So conclusion is, why would we want to make a compartmental model of such a small neuron? And for AWC, since we have this delay because of the very slow velocity of signal propagation, we think this is useful. For RMD, this is not necessary. You could just use a point neuron model and it will capture the bistability, and you won't have to take into account all this extra simulation.
This model was made to replicate a previous point model that was coded in XPP out. This model was coded with neurons, specifically with neuron Python. This model was able to predict and replicate experimental data, and it also was able to replicate channel knockouts. I think that because it has this ability to replicate channel knockouts, it has usage in channel pathologies or ion channel diseases in the future, and may also be useful in a certain topic of C. elegans that is actually prominent in research, where you not only discover, or you not only research loss of function mutations, but you also research gain of function mutations. I think by manipulating conductances to be much higher, or instead of much lower, you could actually study channel pathologies that actually result in gain of function. And of course, bistability could stabilize motor responses against presynaptic noise. By having those voltage thresholds, you can actually stop presynaptic noise from causing a constant state switch. And that's references, and that's it. Thank you.
So I like the way that you put together this into a function, and I want to ask a kind of general question to see if I see it clearly, and stop me if I'm wrong here. But so I want to ask, so you have these channel knockouts, and then you have your interpretation in terms of this Schmidt trigger. How do you think you could put those channel knockouts together to really see the mechanism for that hypothesized function?
That's actually a really good question, because one thing I did not include in this presentation when I was initially doing my exploration of this voltage thresholds, I found that if you injected a very, very precise current, you could actually cause, here, I'll go back to the graph. If you injected a very precise current as to almost confuse the cell if it should go into the state 1 or state 2, instead of having this very clear drop or this very clear rebound, you would actually get it stuck in some kind of very weird transient state. And that plateau could last for almost 50 to 100 milliseconds. And I thought, wow, that would be really crazy if this happened, because if it got stuck in this transient state for 100 milliseconds, that's a large amount of time for a C. elegans to be unable to switch between two states. And I thought that could show a large significance. However, the precision of the current was so precise. It was literally 13.3418755 that I was like, there's no way a C. elegans would ever reach that precise of a current, especially in the presence of noise.
So you were asking about the knockout. There's one channel, which is a calcium voltage gated channel, which is only expressed in RMD, not in AWC. And you see that because of the genetics. If you knock that out, we predict, and there should be no bistability. So it should have only one resting potential. If you do an experiment where you can knock that out, and it's hard to do these experiments because they're really small neurons, so you'd have to knock it out on one neuron, and it's hard to say if you're knocking out the whole network. But if you do it, you should observe loss of bistability, and that should compromise some kind of motor function.
Could you imagine some type of dread approach or something like that?
Oh, with chemogenetics? Yeah. If you were to use chemogenetics in this case, I think it would actually work. There's been already a lot of cases of using GCamp already in C. elegans, as seen from the references. If you were to use chemogenetics to control those channel openings, yeah, I think it's certainly possible, especially to cause one C. elegans to go between a state where it can't be bistable and one where it can.
You may also be able to look for cases where you actually see freezing behaviors. Maybe there are freezing behaviors in the control behavior.
Yeah, that brings me to another point. We would have loved to have more biological reference data for a lot of these knockouts. However, of course, in a computer, changing these knockouts is just changing conductance to 0. If we had a lot more reference data, I think a lot of these would have a lot more meaning, especially with some of the other phenomena we observed.
Well, there's lots of data with C. elegans swimming and doing behaviors and things like that. So it's kind of it's a place where you could try to point something out from your simulations and then look further.
Absolutely.
I have a comment on this slide precisely. So you actually say that the knockout of this CAV3 removes the bistability. But in the previous slide, where you had a different current,
It's the blue line.
Oh, okay, no,
Yeah.
it just didn't go up. Okay, yeah, okay.
It actually just never reaches bistability. Which is why, I originally got this graph and I was like, well, how do I know if it's not?
No,
Yeah.
okay, no, I thought it was the other way around. Okay.
Yeah.
Okay, thanks. Then it's
The point of the was injecting with so much current to see if it could be bistable. Oh.
But in the random, in the stochastic regime, you still have it. No? With this crop out.
No, actually, if you look at the scales, these scales are like -10 to 20, but this scale is, I'm sorry, it's small, but it's actually like -80 to 70. It never goes up. The
Okay, no.
bottom left one, yeah.
Okay, it's fine.
Bottom left one is CAV3.
Just a zoom on the lower line. It's not continuous.
Yeah, I should have kept the scales the same.
Yeah, okay, that's fine.
Thank you. Oh, never mind. I the school with a large biology, but also a little bit of data science focus. Coming to the school and really having the extra, the next step of data science to have computational neuroscience, it was very fun to actually be able to model some of the things that you can see in real life. And it was good to have the ability to model data and analyze that data and then ask yourself, okay, could I see this data in real life? And it's a kind of loop where, oh, I see this data in real life, I can model it again, oh, that mimics real life data, and you can just keep going and going until you get more and more precise and realistic data. I thought that was good.
I study physics, so I knew only high school level neuroscience. So I learned a lot, both about the Hodgkin-Huxley model and how it can get really complicated when you go to an actual animal and you see all the channels. And I didn't know almost anything about C. elegans, and now I know a reasonable amount, and I can see this can be extended very far if you take other neurons, and if you really need more data on the different neurons and electrophysiology. But this is really extensible if you go forward and try to investigate specific circuits. And yeah, so I learned a lot about biology.
Hello, hear me? So, here? Okay, so we are going to present our project, bursting analysis in the HH model in response to potassium channel related in pituitary corticotropes. And well, we all know the Hodgkin and Huxley model that integrates the primary plasma membrane ionic currents found in pituitary corticotropes. It includes four main ionic currents. The L-type voltage sensitive calcium current, a 18T type voltage sensitive calcium current, a voltage sensitive calcium current, and a non-voltage sensitive calcium activated potassium current. The remaining current represents all the other ionic contributions. So under specific parameters, the model demonstrates a different form of bursting behavior. Well, here we can see all the currents, the equation that we already know, and here is the bursting pattern. These examples are from persistent sodium current that induces repetitive action potential firing in the hippocampus and burst firing of substantia nigra dopamine neurons. So this bursting phenomenon arises from the coexistence of a fast time scale linked to the membrane action potential and a slower time scale associated with the gradual action of the calcium in the endoplasmic reticulum. There is bursting directly driven by slow modulation of the calcium concentration in the endoplasmic reticulum. And potassium, as calcium has a slow behavior, we were interested in potassium that is also considered as slowly activated voltage dependent current. So we intend to explore if calcium concentrations is involved in the modes of bursting transition using the Hodgkin and Huxley model. So here we are showing some examples developing animal models with different electrophysiological techniques from depression and for depression, anxiety, and for different anatomical regions and epilepsy. So we used mainly NumPy, the Hodgkin and Huxley model, and we modified the condition of the applied current J to observe the effects on MNH, and this can help understand how the potassium channel responds to different input conditions. And by this, we intended to determine if bursting behavior is possible, by observing these parameters. So we started with our initial fixed parameters, and we started to change the current injection. We started with J equal to 2, and we found the action potentials and its relation with the injected current. We also analyzed the M variable jointly with the action potential, with the appearance of the action potential. So here we can see M very small in this very small slope, and wherever is the EM, it's the action potential that is firing. And we did it for several J intensities. And we analyzed also the amplitude of M that is pretty fixed. And its association, well, this continues to be with different J intensities. Then we were interested also in observing the behavior of the H parameter that is, typically the opposite of M. And we can see this transition over here, that they have the opposite behavior. We also did this for, a wide number of intensities. And in this slide, we see the extreme cases where J equals 1 has no activity. We see M and H, and this is like nothing. And then with J equals 630, we see an action potential, but it has like, non-conventional shapes. And still we don't have a burst until.
Hi, my part of storytelling and write the code. And our first idea is working with busting in xenon potassium, but it was a bad idea. But we described it yesterday because all the week I write the code. All the week I tend the values of gate M, gate 8. Oh my gosh, where is burst? Where is burst? I don't find. And all the moment, Adriana, I'm crazy. I don't find bursting. But Mauricio told me yesterday, Sui, I think you don't fight bursting. Oh my God, now. And we have different condition for J. It's a principal input, and we have different values of membrane protection, and we have the frequency of the membrane protection, fire of membrane protection, and here, oh my gosh, also we found here all the week the bus, and we have bus, but this is this it doesn't but of Hodgkin-Huxley, because yesterday we discovered that potassium channel need slow and fast current, and it doesn't have. Now we have another model based in the Hodgkin-Huxley where we to find a burst. It's just well. Thank you. Sorry, my epopeia and about bursting, it's I'm crazy. Our conclusion, the study of the potassium channel, it's important when we are trying to understand the dynamics of reverse potential, but for burst studies are better to look in the calcium channel. The bursting activity of neuron has been described in terms of the qualitative slow fast theory. And what to say? We want another moment to study a transition between tonic spike or burst in the calcium channel. Thank you.
Yes. We were based on a previous study that modulated three types of currents, calcium currents. I don't remember exactly which one six, but we tried to do a similar methodology, and we couldn't get the bursting activity.
We could to find current another model when we compare the Wind-Matthews model and the Hodgkin-Huxley model. The difference it's about the variable, because the Wind-Matthews model has a three variable, three dimension.
Well, yes. Well, I studied the Hodgkin and Huxley model for the first time a long time ago. So for me, it was very nice to see it implemented in, well, code and to play with the parameters. I mean, that's very nice to do because in a real cell, it's very difficult. I come from a biological background, so the whole school for me was, well, I learned a lot, and I intend to implement some of the new knowledge that we have received here. So this was an excellent exercise for me to start well playing and going through the code.
No results, it's result. And I learned in Hodgkin-Huxley because I working with map-based, and I work with synchronization. It's different for me, the dynamic, the model, and I work about the potassium channel, calcium channel, because it's new for me.
Negative yeah.
Don't leave, don't leave. T-shirts, t-shirts, t-shirts.